mechanism, enzyme mechanism can be determined through radioisotope exchange. So these chapters or these techniques is useful for understanding the catalytic mechanism of the enzymes. This method involves the use of radioisotopes. So which are the isotopes of the elements that emit radiation? So emission of the radiation through the isotopes, and it can measure. So for that you have to first specifically label that atoms or a functional group within the enzyme molecules. So various techniques are in the markets, and correction techniques is also there. So researcher gain by monitoring this exchange of radioisotope between the enzyme, substrate, product or other molecules. Anyone or researcher can gain insight to know this exchange of radioisotope involving in enzymatic reactions. So first of all, the first steps involves introduction to radioisotope labeling. So generally two types of radioisotopes are used, and that is incorporated at specific position within the enzyme molecules. So one is tritium 3H or carbon 14 or 14 carbons. These carbon 14 are radioisotopes emits a radiation which can be detected and quantified by using specific equipments. I will also give you a name of that also in the another points, where we will study the measurement of radioisotope exchange. The label enzymes or label molecules is incubated with the substrate or other molecules involved in the reactions, enzyme substrate complex. So as a reaction proceeds, this labeled atom or a labeled functional group may undergo exchange with the non-labeled atoms or its surrounding environment or surrounding medium. The rate of radioisotope exchange can be measured by periodically you have to take a sampling. And after taking a sampling of the reaction mixture and analyze it through this technique. For example, liquid scintillation counting. This liquid scintillation counting, I will also explain radioisotope techniques, various radioisotope techniques. I had already explained during the intermediary metabolism. Third, interpretation of radioisotope exchange data. So, in that things, the rate and pattern of this exchange provide a valuable information about the mechanism of enzyme catalysis, enzyme actions. For example, if you are labeled atom exchange rapidly with non-labeled atom, labeled atom exchange rapidly with the non-labeled atoms, what it suggests? It suggests that this atom is involved in first step of the reaction. On the other hand, if the exchange is slow or very negligible, what it indicates? It indicates that the atom is not directly involved in any catalytic reactions under the study. Similarly, When we studied with the enzyme or substrate complex, you must know, as I said in the book, enzyme is nothing. It has a site, two sites, active site and binding sites. These active sites, identification of active sites through the radioisotope techniques. So by selecting a specific amino acid residue within the enzyme active site, one can determine the residue which are directly involved in the catalysis. For example, if labeled residues exchange rapidly with the non-labeled atoms, it suggested that this residue participate in the chemical transformation of the substrate. Now you read 4.
By
Identity.
identification of active site residue. By selecting labeling specific amino acid residue within the enzyme's active sites, researcher can determine which residue are directly involved in catalysis. For example, if a labeled residue exchange rapidly with non-labeled atoms, it suggested that this residue participates in the chemical transformation of the substrate.
Okay. Now you have to remember only the example if level residues, radioisotope level residues can exchange with the non-level atoms or not. If it is so, then participation in the chemical transformation of the substrate can possible. Now same investigation of the reaction intermediates. So radioisotopes can exchange also provide insight into the formation and stability of the reaction intermediate. So these words you have to remember. Insight. It provides insight in the formation and stability of reaction intermediates. By monitoring the exchange of label atom or a functional group during different stages of the reactions, researcher can infer the presence and the properties of transient species that are difficult to detect by other means. So you can measure the properties of transient species which cannot detect it by another means. By the means of radioisotope, you can label exchange or monitoring of these label exchange material into the functional group or during the different stages of reaction it goes or not. Now sometimes another applications in the radioisotope for the study of enzymatic mechanism, one can also compare a different enzyme variant. What type of different enzyme variant or a mutant? So radioisotope exchange can be used to compare or monitor the catalytic properties of the different enzyme variants or a mutant. So by labeling a corresponding position in the different enzyme variants and measuring the radioisotope exchange rate, one can identify the key residues or the key region that contribute to observe differences in the catalytic activity. So overall, I can say radioisotope techniques can be useful for determining the enzyme activities, rate of enzyme reactions, as well as from substrate to product or substrate to intermediate reactions. You can detect the step also. These techniques is also useful for determining the metabolic pathways. So limitation and challenges. These are the limitations and challenges of radioisotope exchange. As you know that it is a powerful techniques, but it requires for studying these enzyme mechanism, it has a certain limitations. So incorporation of radioisotope into atoms or functional groups or active sites It requires technical knowledge. So technically challenging matter is involved in that. Another is specialized labeling method you should know. Otherwise it cannot be possible. Additionally, after the labeling and doing this reaction, you should know the interpretations. In what type of interpretation of radio exchange data from substrate to functional group to intermediate group to end product. So these exchange data require careful considerations of factors. Which type of factors are involved in that? Same as factor affecting enzyme activity. So here you saw you have which solvent is accessible for labeling, which solvent, as well as which protein or their dynamic structures, which functional group is attached with that labeling, whether this label confirmation is possible with the end product or not. So potentially artifacts arising from the labeling procedure. In nutshell or in conclusion, I can say these chapters or this chapter name is Mechanism, Enzyme Mechanism Determination by Radioisotope Exchange is a valuable tool for investigation the mechanism of the enzyme by proper selecting the enzyme specific atoms or a functional group within the enzyme molecules and monitoring the exchange between the labeled atom to non-labeled atoms, and one can get insight or steps involved in the enzymatic reaction as I involved 1 to 6 steps. So this is all about the radioisotope techniques useful for